next speaker is Dr. Julieta Orlando from Universidad de Chile and also from the Millennium Institute BASE in Chile with the talk Microbiomes at the End of the World from a Community to an Intra-Population Perspective. Please, Julieta.
Can you listen to me?
Yes, we can listen and see your presentation.
Thank you. Okay. Good morning to everyone. Thank you for the invitation to the organizers of this meeting. In this opportunity, I want to show you this is not working, that. A general presentation of the Millennium Institute BASE, an overview of some of our research related to microbiomes, and some of the outreach activities we perform. The Millennium Institute BASE is focused in studying the biodiversity of Antarctic and subantarctic ecosystem and is funded by the Millennium Initiative Program of the Chilean Research and Development Agency for 10 years. When we submit our proposal for the Institute for the first time, the International Committees on Biodiversity and Climate Change jointly published a report warning about the importance of putting nature at the center of decision-making. On that occasion, we thought that a promising territory to address this challenge was Antarctica and the subantarctic territories, not only because of our experience studying its biodiversity for some time, but also because it is a territory with relatively low anthropogenic impact and drastically affected by increases in temperatures, among other changes. Therefore, we propose 4 objectives that define our research proposal. First, we propose to study the influence of past climate changes on the evolution and shape of the Antarctic and subantarctic biodiversity. Second, to predict the fate of this biodiversity, including shifts in native species distribution, extinction risk, and exotic species establishment. Third, we aim to establish a long-term biodiversity monitoring system. And finally, we pretend to design management proposals to protect and conserve the Antarctic and subantarctic biodiversity. So, through 8 research lines, we investigate about the past, present, and future of the Antarctic and subantarctic biodiversity to contribute with knowledge to decision-making in terms of conservance and governance. This associative research proposal brings together researchers from different disciplines and institutions from Santiago to the south, with a total of 25 researchers, including main, associate, and senior researchers. Our host institutions are Universidad de Chile, Pontificia Universidad Católica de Chile, Universidad de Concepción, Universidad Austral de Chile, Instituto Antártico Chileno, and Universidad de Magallanes, which give us the opportunity to have researchers from Santiago to Puerto Williams in Navarino Islands. The research we are carrying out will not be possible without the participation of a huge group of students, both undergraduate and graduate, from different study programs and with diverse experience. Along with them, a group of postdoctoral and adscript researchers collaborate with the achievement of our objectives. Notoriously, we have a dominance of young women researchers. Antarctic and subantarctic biodiversity is much broader than many think, including various organisms, among which, of course, are microorganisms. Although there are a lot of study models, we have ventured into the study of the microbiome of various environments, which is what I want to show you today, along with the work that is currently in progress. To start, I tell you something about our work studying the lichen microbiome. The first step in this research line was to know which peltigera lichens, our study model, inhabit the study sites. In this regard, we contribute with reports of representatives of this genus in southern Chile and Antarctica, and the latest product consists of a database with more than 1,000 specimens identified and published on the Global Biodiversity Information Facility, GBIF. And interestingly, some of the peltigera lichens we found are endemic of the south tip of southern South America. Then, using a specific lichen species, Peltigera frigida, we try to determine the origin and selection of the lichen microbiome. For that, we studied the bacterial communities associated to thalli and substrate of this lichen species. And we found that some bacteria will be dispersed together with lichen propagules, for example, Sphingomonas, which are abundant in the lichen thalli, but not detected in their substrates. In addition, we found some taxa that could be negatively or positively selected from the substrates, since their abundances were very different when comparing thalli and substrates. Currently, we are focused on 4 species of Peltigera lichens, 2 globally distributed, Peltigera fusco-praetextata and Peltigera rufescens, and 2 endemic of southern South America, Peltigera frigida and Peltigera antarctica. With the participation of students of different levels and programs, we are interested in compare the microbial diversity and structure, the microbiome functions related to, for example, nutrient acquisition and hormones production, and the search of new potential symbionts such as basidiomycete yeast. Moving on the microbiome of penguin soils, first, we studied through 16S metabarcoding the bacterial communities in soils from the Cape Shirreff in the South Shetland Islands, impacted by marine birds and pinnipeds. We found that the soils impacted by the penguin colonies were the most different from the control, with nitrate being the most influential edaphic factor. So next, we delve into the bacterial diversity in these soils through metagenomics, including an additional point in the South Shetland Islands. And we assembled 100 mags and searched genes potentially involved in nitrous oxide emissions, finding that the relative abundance of genes related to the nitrification was at least double in soils impacted by birds than in soils without animal influence. It is pending the measurement of the nitrous oxide that is actually released from these soils, but these soils could be an important source of this potent greenhouse gas. Due to the importance of penguins in impacting the soil microbial diversity, we sampling soils impacted by these birds along the Antarctic Peninsula, and currently we sequence more than 50 metagenomes, and we have more than 1,000 mags already assembled. We are searching for differences among localities and penguin species, and preliminary results reflect that the penguin species of the genus Pygocelis seems to be the most influential factor on the microbial diversity in these soils, independent of the location of the sampling sites. In addition, we are predicting microbial functions related to greenhouse gas emissions and the ability of soil microorganisms to biodegrade different pollutants such as fuels and microplastics. The last research line that I want to share with you is the one related with the microbiome of marine animals. In this line, first we compare the microbiota associated with the gut tissue of the sea Archinabatus agassizi with the microbiota in the gut content and the sediment where these animals live. And we found that the microbial community in the gut tissue is less diverse here and different from the other two. In addition, we found key taxa with potential critical functions for the host, spirochetes able to fix nitrogen that might benefit the host in a context of low nitrogen food, and sulfate reducers that could contribute in degrading recalcitrant organic compounds present in the sediment. Then, analyzing the key OTUs previously detected in the communities, we performed microdiversity analysis to study the intra-population phylogeography of bacterial taxa across the Southern Ocean, and we found three main patterns. First, a genetic homogeneity within the Patagonia province. Second, a barrier to dispersal between Patagonia and Maritime Antarctica due to the Antarctic polar front. And third, a connectivity between Kerguelen Island and Patagonia or Maritime Antarctica due to the Antarctic circumpolar current. We are currently expanding the study models, including marine sponges, limpets, and fishes, among other models. And in each case, we are searching for potential contributions of the microbiome to the host physiology. And we are evaluating coevolution and phylosymbiosis signals between hosts and their microbiota, as you can see here illustrated in the confrontation of the phylogenies of Arpagifer fishes and a dominant OTU of its microbiota associated with Alivibrio bacteria. Finally, we are assessing the impact of multiple stressors such as temperature, pH, and salinity on the diversity, structure, and function of the microbiota of different hosts. I don't want to end this presentation without briefly showing you some of the initiatives we carry out regarding outreach. As researchers of the Millennium Institute BASE, as you can see here, we have the opportunity to contribute with talks, notes, and interviews in various in-person, virtual, and press media at both national and international levels. And one of the most important outreach activities this year was our active participation in the Science Festival organized by the Chilean Ministry of Science, being present in the regions of Magallanes, Los Ríos, Biobío, O'Higgins, and Metropolitana in Chile, with the collaboration of more than 30 volunteers. Finally, I want to leave you the links to 2 examples of free access material that you can find on our website, and I invite you to follow us on our social networks on Instagram, Twitter, or X, and Facebook to stay informed about our progress. Thank you very much for your attention.
Thank you very much, Julieta. Perfect time. I want to acknowledge all our speakers from this part of this session.